We'll continue the lecture talking about osmoregulation and excretion. So how does an albatross drink salt water without ill effect? So we'll see in these lectures how he tried to manage with this problem. But first we need to understand some definition. So osmoconformers are some marine animals are isosmotic with their surroundings and do not regulate their osmolarity, while osmoregulators expend energy to control water uptake and the loss in hyperosmotic or hypoosmotic environment. Tenohaline, most animals cannot tolerate significant changes in external osmolarity, while ureahaline, animals can survive large fluctuations in external osmolarity. So the concentration of water and solute must be maintained within narrow limits. Osmoregulation regulates solute concentration and balances gain and loss of water. Like in freshwater, animals show adaptations that reduce water uptake and conserve solutes, while in desert and marine animals, they face dehydrating environment that can deplete body water. So excretion is getting rid of nitrogenous metabolites and other waste products. The first concept is osmoregulation, balance uptake and the loss of water and solutes. The osmoregulation is based largely on controlled movement of solutes between internal fluid and the external environment. Cells require a balance between uptake and loss of water. The osmolarity, the solute concentration of a solution, determines the movement of water across a selective permeable membrane. So water moves from a high potential into low potential water. In other words, water moves from diluted solution into concentrated solution. If two solutions are isosmotic or isotonic, movement of water is equal in both directions. If two solutions differ in osmolarity, the net flow of water is from the hypoosmotic diluted to the hyperosmotic, which is the concentrated solution. So we can see in this figure how is the net movement flow. As you can see from the arrow, the water moves from the diluted solution into the concentrated solution, from the hypoosmotic side into the hyperosmotic side, from the lower solute concentration into the higher solute concentration, from the higher free water concentration into the lower free water concentration, and this through a semi-permeable membrane. So in marine animals, most marine invertebrates are osmoconformers. Most marine vertebrates and some invertebrates are osmoregulators. These, for example, marine bony fishes are hypoosmotic to seawater. They lose water by osmosis and gain salt by diffusion and from food. They balance water loss by drinking seawater and excreting salts. As you can see in these figures, the osmoregulation in marine fish, you can see that the gain of water and salt, they take salt ion from the food. The dark arrows are for salt and the white arrows are for water. So you can see that water and salts are gaining from food and seawater. While there is excretion of salt ions from the gills, there is excretion of salt ions and small amounts of water from urine from kidneys through urine, and most of the excretion occur of salt through gills, and there is osmotic water loss through gills as well and other parts of body surface. In case of freshwater animals, they constantly take in water by osmosis from their hypoosmotic environment. They lose salts by diffusion and maintain water balance by excreting large amount of dilute urine. Salts lost by diffusion are replaced in food and by uptake across gills. So you can see here from the figure that most of the water and some ions are taken up through food. While the uptake, there is as well uptake of salt ions by gills and uptake through water from gills. So the osmotic water gain through gills and other parts of the surface. So actually it's basically the other way around from the marine. Excretion of salts and ions, there is a large amount of water are excreted in urine. So you can see that the major difference here that some of the excretions or the release of ions or water in marine animals are through gills, which is not the case here in the fresh water. Looking closely for how birds regulate their salts in digestive body and osmoregulation, an example is found in the nasal glands of marine birds, which remove excess sodium chloride from the blood. You can see here that this marine bird had a nasal salt gland with a duct that opens near the nostril with the salt secretion. This is the location of the nasal gland. So to be familiar, a blue arrow will be the salt movement and the red arrow will be the blood flow. So salt is transported from blood into secretory tubules which drain into central ducts leading to the nostrils. So basically they release the salt from the nostril. If we enlarge if you enlarge the nasal gland, we'll find that there is a lot of blood capillaries and there is a duct which is a secretory, which leading to the secretory tubules, and these capillary and secretory tubules which convey the salt until reaching to the external via the nostril. If you look closely inside the secretory tubules, you can find there is two pathways which are opposite to each other. The red one for the blood flow and the blue one for the salt movement. As both ways move, there is transport of the salt from the blood into the lumen of the secretory tubules. So exchange occur due to the counter current movement. So animals that live in temperate water, some aquatic invertebrate in temperate water and temperate ponds lose almost all their body water and survive in dormant state. This adaptation is called anhydrobiosis. Animal like this, you can see the tardigrades. This is a scanning electron microscope image. So they inhabit the temporary bonds as well as droplets of water in soil and on moist surface. You can see that it can exist in two forms: hydrated tardigrade and dehydrated tardigrade to conserve the loss of water. An adaptation called anhydrobiosis. For land animals, adaptation to reduce water loss are key survival on land. Body covering of most terrestrial animals help prevent dehydration. Desert animals get major water saving from simple anatomic features and behaviors such as nocturnal lifestyle. Land animals maintain water balance by eating moist food and producing water metabolically through cellular respiration. So you can see here how what is the factor that control the water balance in two organisms, the kangaroo and the human. The kangaroo needs around 2 ml per day, while human beings need around 2,500 milliliter per day. So most of the water comes to the kangaroo through the derived from the metabolism, while in human most of the water is ingested in liquid, while the other portion is ingested in food, and the minority is derived from metabolism, and that's a major difference between kangaroo and human. In case of the losing water, most of the water is in kangaroo is lost via evaporation, while in human most of the water is lost via urination. Energetics of osmoregulation. Osmoregulators must expend energy to maintain osmotic gradient. The amount of energy differs based on three factors: how different the animal's osmolarity is from its surroundings, how easily water and solutes move across the animal's surface, the work required to pump solutes across the membrane. Second concept, an animal's nitrogenous waste reflect its habitat. The type and the quantity of animal waste product may greatly affect its water balance. The nitrogenous breakdown products are most significant waste of proteins and nucleic acids. The forms of nitrogenous waste are either ammonia, urea, or uric acid. The ammonia animal that secrete nitrogenous waste as ammonia need access to lots of water. They release ammonia across the whole body surface or through the gills. While urea, in case of urea, liver of mammals and most adult amphibians convert the ammonia to less toxic form, which is urea. The circulatory system carries urea to the kidney where it is excreted. The conversion of ammonia to urea is energetically expensive, so excretion of urea requires less water than ammonia. The third form is the uric acid occurs in insects, land snails, and many reptiles, including birds, mainly excrete uric acid. Uric acid is relatively non-toxic and doesn't dissolve readily in water. It can be secreted as a paste with little water waste. That's why most of the animals that try to conserve the water, they urinate in the form of uric acid. Uric acid is more energetically expensive to produce than urea. So we can see here that either proteins and nucleic acid, when they got used by the body in the form of amino acids or nitrogenous weights, the amino group present in these compounds, in most aquatic animal, including most bony fish, it will be excreted in the form of ammonia, while in most mammals and most amphibians, sharks, and bony fish, it will be released out in the form of urea, while in most reptiles, including birds, insects, and snails, which needs more conservation of the water, they are released or excreted in the form of uric acid. So concept 3, the diverse excretory systems are variation on a tubular theme. So basically, all of the excretory system throughout the organism are a form of complex tubular structure. Excretory systems regulate solute movement between internal fluids and the external environment. So the excretory process can be divided into four other processes. Most excretory systems produce urine, which is a filtrate, by pressure filtering body fluid and then modifying the filtrate content. In the figure, you can see it's a tubular structure where the blood capillary moves until entering a capsule, which we will know now it's called Bowman capsule in case of mammals. And this from this blood, there is a process of filtration where the blood is filtered. So the filtered now, which will form the urine, will move in the excretory tubule until forming the urine to be excreted. So the key function of most excretory system, first thing is filtration. Actually, it is filtering the body fluids from water and solutes, and all of this filtrate is collected inside based on the pressure of the blood. So that's why any disturbance or abnormality in the pressure of blood will straight away affect the function of the kidney. The second process is reabsorption. In reabsorption, there is a reclaiming of important and valuable solutes instead of being wasted and lost in the urine. In the third process is the secretion. Other substances such as toxins and excess ions are extracted from body fluids and added to the content of the urine. Last process, which finally is the excretion, that means the processed filtrate or urine after reabsorption and secretion and filtration are released from the body. So if we look to a survey of the excretory systems through different organism systems that perform a basic excretory function vary widely among animal groups. They usually involve a complex of network of tubules. So you can see the first, the primitive type of a kidney is called the protonephridia, and this protonephridia is present in an animal, a very primitive kind of a worm called planarians. And you can see it's a network of dead end tubules, which in the figure called flame bulb, consist of a capsule and the tubule cells. And these protonephridia, from the whole mount of the planaria, you can see they are arranged laterally throughout the body of the worm. The smallest branches of the network are capped by a cellular unit called the flame bulb. These tubules excrete dilute fluid and function in osmoregulation. So these flame tubules are an example to collect the filtrate and open to the outside through the body wall to release the excretion or the filtrate. Another type of excretory system is called metanephridia. Metanephridia, actually the main difference that these metanephridia exist in worms like the earthworm, and you can see it is repeated over each segment of the worm. And when you look closely, you can find that each segment of the earthworm has a pair of open-ended metanephridia that open to the outside by external opening. Metanephridia consists of a tubule that collect coelomic fluid and produce dilute urine for excretion. And you can see from the figure, this is a component of the metanephridia. The other type of excretory system, which exists in insects, for example, and terrestrial arthropods, the Malpighian tubules. The Malpighian tubules remove nitrogenous waste from the hemolymph and function in osmoregulation as well. So from the figure, the enlarged part of the insect, you can see that Malpighian tubules located mainly in the midgut around this, just below the stomach of the grasshopper. And this Malpighian, which looks like the hair outgrosses the structure, actually it regulate and excrete and regulate the amount of salt water and excrete nitrogenous waste until collecting these waste product through the rectum, where there is the process of reabsorption of valuable elements, valuable compounds like water, ions, and organic molecules. These Malpighian tubules are out-pocketing of digestive tract that remove nitrogenous waste as well and function in osmoregulation. Insects produce a relatively dry waste matter, as we said before, mainly uric acid. Some terrestrial insects can also take up water from the air. So if we look to the most advanced excretory system, so it's in human. So the kidney, it's actually the excretory organ of vertebrates and function in both excretion and the osmoregulation. So in the first figure, you can see where is the kidney is located. When you enlarge the kidney, you can see that the kidney is supplied by renal artery and renal vein, and there is a ureter coming out of the kidney for collecting the urine. Generally, the kidney is divided into a cortex, the outside layer, and the medulla, the inside layer. We can take a section from the kidney looking inside to see what is composed of. When you take a section in the kidney and look inside, you can see that there is different types of nephrons, which is the basic unit of the kidney. You can see that the renal cortex is outside, the renal medulla is inside. You can see that there is two types of nephrons, which is a cortical nephron and the juxtamedullary nephron. Looking closely into the nephron structure, you can see that the nephron organized from different regions, and all of them are supplied by blood vessels for the sake of the absorption and reabsorption and filtration. Generally, the blood vessel enters the Bowman capsule and comes out, and the Bowman capsule do the first process is filtration, collecting the filtrate, and then reabsorption, secretion, and then excretion. So the general parts of the nephron are first is a Bowman capsule, followed by the proximal tubules, followed by the lobe of Henle, which actually consists of descending limb and ascending limb, then the distal convoluted tubules or distal tubules, then the collecting duct, which conveys urine to the outside. If we look to the structure to summarize the process, you can see the glomerulars, which is like a tuft of blood vessels. Actually, it's the connection between the arterioles and venules. And these Bowman capsule filtrate the blood, collecting the filtrate, and then the reabsorption occur, the reabsorption for the important and valuable compounds like water and ions and organic molecules. And then from the blood again and from the interstitial fluid, secretion occur to some of the toxins, and then excretion for the filtrate. If we look closely to the concept for is the nephron is organized into for step-wise processing of filtrate. So this is actually the same figure you have just seen. So we'll try to relate the function and the structure of the nephron. So the filtrate produced in Bowman capsule contains salts, glucose, amino acids, vitamins, nitrogenous waste, and other small molecules. You can see where is the location of the Bowman capsule. The proximal tubules, you can see highlighted in figure, reabsorption, in these proximal tubules, reabsorption of ions, water, and nutrients take place in the proximal tubule. Molecules are transported actively and passively from filtrate into interstitial fluid in a process called reabsorption, and then to capillaries. Toxic material are actively secreted into filtrate in the process called secretion. As the filtrate passes through the proximal tubule, it becomes concentrated. While in descending limb, reabsorption of water continue through an important protein called the aquaporine. The high osmolarity of interstitial fluid drive the movement. The filtrate become increasingly concentrated. Then we have the ascending limb. Salt but not water is able to diffuse from tubule into interstitial fluid. The filtrate become increasingly dilute. Then we have the distal tubule. The distal tubule regulates the potassium and sodium chloride concentration of body fluids. Then we finally have the collecting duct. The collecting duct carries filtrate through the medulla to the renal pelvis, and then urine is hyperosmotic to body fluid and got excreted. In this slide, we'll summarize the process that occur for each nephron. So each nephron starts with a Bowman capsule that undergo filtration where water, salts, bicarbonate, protons, urea, glucose, amino acids, and some drugs are filtered from blood to reach the proximal tubules. Be aware of the key. Red arrow is for active transport, that means transport needs energy, and the other arrow, the cyan arrow, is for the passive transport, doesn't need energy. So in the proximal tubules, bicarbonate, sodium chloride, and water and nutrients and potassium are moved out of the tubule, while proton and ammonia is moved inside to the tubule. In the descending limb of lobe of Henle, water is reabsorbed passively. And then in the ascending lobe of Henle, sodium chloride is reabsorbed passively. Again, it will be after that it will be reabsorbed actively. In the distal tubules, potassium and the proton is pumped inside the tubule, while sodium chloride and bicarbonate are reabsorbed actively, while water are reabsorbed passively. Now the filtrate have reached the collecting duct. Each nephron releases its filtrate into the collecting duct, which is soon after that sodium chloride again reabsorbed actively because it's an important molecule for the body. Finally, the urea and water again is reabsorbed passively, and urea is reabsorbed passively. And this concludes the formation of urine, and urine is released. Thank you.